welcome Doug.
Thank you. Cool. Thank you, Tisha. Thank you guys for coming. So what I want to talk a little bit about today is dive a little bit more deeply. Some of you joined us last night for a conversation about science and extinction. I want to explore a little bit of a different theme that is a guiding motif in a lot of the work that I do in both terrestrial and marine systems and in freshwater systems. The general question that motivates a lot of my research is, as I think Tricia will summarized, is how do humans shape the structure and the function of both ecological communities and ecosystems? Of course, that's quite broad. I think it's really fun, really informative, really important to sort of ask those questions in the here and now and sort of modern ecological context to see what we can learn from this modern change that gives us some insight on how some of this human change may have reshaped the history of ecology in these same spaces, and then to begin to try to predict what kinds of change we may see humans engineering in communities and ecosystems going forward. Of course, that's broad. What I want to focus down on here today is a specific set of questions that relate quite a lot, as the title of the talk, to ecological dynamics in a smaller world, that relate very specifically to changing body size in communities. And this interest in changing and shifts in body size of life are inspired, for me, by some very dramatic, very conspicuous shifts in distributions of life in a lot of the systems that we study, we live in, interact with. Here, for example, is a view of what Utah might have looked like during the Pleistocene 15 or so thousand years ago. You have a very different kind of landscape of fauna, right? A lot of things in this picture that look quite unfamiliar to us, right? Camelops, the camel, a short-faced bear, Smilodon, the saber-toothed tiger way in the back, sort of lurking around. Mammothus, these animals that were, if one thing unites them, one thing that unites them is they're not here anymore. Another thing that unites them is that they're big, right? You can sort of fast forward to what a hike might look like on a Sunday around this same ecological space here in Utah, and you can see that things are quite different. I mean, that might change your hike for better or worse. A key difference is that you don't have that space shared with a lot of the larger life forms. We still have some familiar faces, right? There's some coyotes still hanging around that were here in the Pleistocene. There's some raccoons, things like that, right? Otherwise, a quite interesting shift with a consistent theme being a lot of the big things have been lost. To put just a slightly more quantitative face on that pattern, this is just a portrait of mammals in the West, a portrait of body size distribution. What I've done, every one of these little microbars here is a unique species. On the y-axis, I'm showing you its body size. It's a body mass. We have here on this end of the distribution really big things, right? These are our mammoths, our ground sloths, and then barely registering on this size distribution are tiny things, many of them, many of species like insectivores, our shrews, our bats, things like that, right? So I've coded them here in color in two different ways. In this mammal assemblage, we have species in blue that were here in the West during the Pleistocene and went extinct, no longer with us. We have in red species that were here in the West during the Pleistocene and are still hanging on. So if we kick out the species that have gone extinct, look what happens to that portrait of body size, right? That is a profound shift. That's a profound shift in sort of the size structure of life in this very space where we're sitting today, right? And there's no way that that shift in size structure could have proceeded without having really important, profound implications for the structure, the dynamics, the operation of the systems where we're sitting, standing, studying today. You can sort of transform this into all kinds of analogies. If you think about a test food with all sorts of different size atoms and molecules, if you were to go through and kick out all of the largest, let's say atoms, kick all the largest atoms out of that solution, you have just a remixing and interaction of tiny atoms. the chemistry of that system is going to be very different. The dynamics of that chemical solution will be very different. If you think about it as a chess game and you pull all the biggest pieces out of your game, the rules by which that game is played are going to be very different. So I'm very curious about how the rules of this game that is life in this space are different because we pulled off the big chess pieces, if you will. So this is not an old problem. It's not a problem that began during the Pleistocene and ended during the Pleistocene. It's something very much that continues on today. You know, it is not a universal kind of selection criteria, but many people, many humans, and many spaces continue to exert at least partial, sometimes very strong, directed selection for the next largest fauna. This is the last grizzly bear, for example, in California. We have it on our state flag. That is the only place you'll find a grizzly bear in the state of California anymore. Of course, we're familiar with examples elsewhere in North America, the Pleistocene gone in Australia. So we're sort of continue, at least in many systems, to ratchet down the body size distribution in these communities, keeping this a question of contemporary interest. In the oceans, and I will talk a lot about science that engages the implications of body size shifts from marine systems for a very specific reason. In the oceans, this Pleistocene megafaunal extinction that we've been talking about that happened on land really hasn't yet been a thing. But we talked last night, some of you joined us for a conversation about ocean extinction. We have not driven very many animals in the oceans extinct. We are beginning to exhibit very strong selection on big things in the ocean, but they're still out there. The ground sloths, the short-faced bears, the mammoths of the ocean are still out there doing their underwater mammoth things. And so it provides a very interesting test ground to understand how body size shifts as they are being engineered, at least at local scales in the oceans, what that means, how that changes ecological dynamics in communities, how that changes connectivity or dynamics that move across communities to ecosystems. So another good sort of motivator for asking these questions is that, in my opinion, this is arguably the very first human impact we've had at a global level on our planet. We, of course, are coming up with all kinds of new ways to globally impact our planet. But this is an interesting one for me because, again, this is really the first profound presence that we've had on our global ecosystem. Working towards some of these answers, trying to understand what the implications might be for loss of big constituents of ecosystems is challenging, is methodologically challenging in many ways because these things are rare, they use lots of space, they're hard to hang out with. Sort of to get a sense of that, like if you have a manager, as they do, ask you, well, we saw a pretty severe loss of manta rays on our island because they're ending up as bycatch in a fishery. We want to know what that means for an ecosystem. You need to produce an answer for them, but where the sort of rubber hits the road as a marine biologist underwater is you're sitting on a reef trying to collect data. Manta ray comes by, you have 15 seconds that are amazing. You, like 2 of those seconds are burnt going, wow, that's cool. The other 13 seconds you're actually trying to take data, and then it's on to someplace else, right? And somehow you have to stitch all those observations together in a way that meaningfully gets towards answers that answer these questions that managers are putting to you about these shifts in the presence or abundance of these large constituents. I often think about the problems for trying to do what I sometimes think about is macrobiology, and not too dissimilar from the problems of trying to do microbiology. If you're studying something really big, you need new techniques, sometimes new tools, sometimes new technologies that help you get your head into the world of these big critters, the same way that we need a whole lot of specialized techniques, tools, microscopes, electron scanning microscopes, et cetera, to get our head into the world of the smallest parts of biological systems. A quick sampling of the kinds of tools that can get us into those worlds and get us some of the answers we need back from these systems. One that we'll rely relatively heavily on in a couple of these science stories are stable isotopes. Grabbing a piece of tissue for that manta ray as it comes by. You feel like a jerk when you do biopsy manta ray, but it's for the greater good of science. Locked in that tissue of these manta rays are chemical ratios that tell you something about the space that this particular animal would use and about what it's eating in that space. So that is very informative. And you can use new kinds of sensing technologies We use, for example, new kinds of sonar technologies that help us get a read on abundance and ecological presence of things like manta rays, and we've done this for sharks and sea turtles, that help you get underwater for longer periods of time. I have 15 seconds maybe in that interaction during a 1-hour dive on a reef. I can stick this sensor underwater for a month at a time, which helps it be a robo-diver that gives me a lot more data. When I do go slumming on land, there are a lot of other really cool technologies that you can bring into play for getting good information about large-bodied species in their ecosystems from terrestrial systems. The great thing is they're hidden under this great lens of water, right, which is the grand problem for doing work, doing ecology under the oceans. This is a sampling of neat technologies that we have at play. One of my graduate students is doing some really cool stuff in Serengeti. She's out there right now, where we have this traditional insight that's being derived from collaring animals. So you get the story of a single individual, for example, wildebeest in the Serengeti, that tells us what space does it use, what does it eat, how does it interact with barriers, with roads, with heterogeneity and productivity. But that tells you the story of one animal. You know, if you sampled one person in Salt Lake City, or 7 people in Salt Lake City, that would tell you a very small subset of what the sort of mean needs are for the community, right? And so that analogy holds to some degree when you're trying to get a sense of what are the mean ecological needs of a population that is, in this case, wildebeest, over 1 million individuals. We can layer that on top of these new sort of macro technologies. Drones, an obvious one, that help us begin to not just observe one individual, but observe hundreds of individuals. Satellite sensing technologies, some of the really exciting ones, allowing us to look down on the planet with resolution into space that gives us a view of about just under half a centimeter, often down to about half a meter. That'd be pretty amazing. And pick out the nose hairs on a wildebeest, but not quite that good. Half a meter, sometimes close to 0.3 meters with these new Earth observation satellites. With those above, in a tiered fashion, above animals that we're getting information from collars, information from drones, we go from individual observations from one animal to hundreds of animals to hundreds of thousands of animals, sometimes synchronously in a landscape. You can imagine the nestedness of that information could be very powerful for telling the story of a wildebeest. And here's just an example of like what happens when you start digitizing the wildebeest migration. This is drone imagery that we're doing, auto tracking of specific individuals, each one of them numbered as they transit a landscape. So there's a whole lot of good information about social biology, about the ecology interactions with species and landscapes that you can extract out of these exciting new tools for looking down at big animals and big systems. The last tool, I'll show you briefly some applications of simulation. You get these like staccato insights for manta rays as they come and go from your marine biological life. If you can stitch them together in a way that creates parameters that go into a simulation that then builds, you can then build insight that takes you towards conclusions that are much more synthetic and interpretive than those little bits are. You put the bits together and then you have some simulation glue that makes a puzzle that starts to give you a picture of what's actually happening in space. So, tools. How about some science? The 3 stories I want to tell you are work that I'm doing about how, or have done mostly, about how loss of large animals and changing body size influence structure of communities, particularly how large animals create connectivity that shapes structure. Then I'll talk about ecological uniqueness and functional uniqueness. Are these big things that we're losing, these big particles, replaceable, or are they fundamentally ecologically unique? Lastly, nutrient distribution. Do this loss of these big particles, these big pieces on the game board of life, do they reshape ecosystem ecology, the distribution of energy, nutrients, resources across the game board that is life? So the first story about influences on structure, we're inspired by portraits of biomass structure. There are lots of ways to paint a picture of structure in a community. These are portraits of just biomass, how it stacks up in a coral reef in the Pacific. The interesting thing is that you get very different answers when you go to different marine spaces that have differing presence of humans. This is a heavily inhabited, heavily fished island. Just down the chain is a lightly fished, less inhabited island, and then you end up at this National Wildlife Refuge, U.S. National Wildlife Refuge, that has nobody except scientists and a lot more fish. So these are just assembled when you drop into a reef. You do a classic inventory of reef fish. You translate those into different trophic guilds. We have your piscivores, your lower level carnivores, your herbivores. You bin them, you add up the mass in these different groups, and you get these biological portraits for biomass. Again, it's just one view of structure in this place. I have to say, I've talked to a lot of you guys, grad students that are doing work with electrofishing. Man, am I envious. If I could electrofish a reef, that would give me so much more insight as to what's out there. If I could electrofish, we'll get to hippos in a bit. If I could electrofish rivers, wouldn't. I think the hippos are very upset about that, but we use what techniques we can to build these portraits, and this is in the ocean done through visual observation. Okay, so there's something interesting about the structure of these unfished reefs, namely that there's a lot of blue sitting on top of that community that isn't found in the unfished communities. That blue are big predators. Those are sharks, jacks, grouper, things like that. Very large, often predatory fish. So, as I say, this is National Wildlife Refuge, so it seems to be a kind of interesting anomaly in terms of the abundance of these large predatory fish. There are lots of interesting questions about what it means to have so many predators in terms of top-down forcing and the dynamics of these communities, all kinds of interesting things that you can do, some that we have done. For example, trying to cage them out from sections of reef and figure out what happens when they're not there. The stories that I want to tell, again, that I want to tell you now, are mostly connected to structure, and in particular to again, this structure. And I think of this as a pretty weird structure, right? This distribution of biomass in this unfished, in this pristine reef. I say weird because it presents some complications for the way that we classically think about biomass being distributed across trophic levels in a community. Our classic view, sort of the Eltonian view of how biomass ought to be distributed, is that it should conform to a roughly pyramidal distribution. You have more herbivores, more prey fishes, and you have less predatory, first level predatory fishes, and then you have less apex predators. And that, of course, that pyramidal structure arises from trophic transfer inefficiencies, classically measured at about 10% of the energy or the biomass, rather from one trophic level moving up to the next trophic level. Of course, we know that there's a lot of variability around that 10% figure, so if it's lower, sometimes it's higher. But anyway, it makes sense that there's a lot of energy that's lost in these transfers, and that loss gives you a pyramidal shape to a community. So what's weird is at these systems like this National Wildlife Refuge, this Atoll Palmyra, is that when you sort of take that plot with all that blue, you end up with something that looks a little bit more like this, an inversion on that classic hypothesis. An inverted trophic pyramid, in fact, is what some people call this. And of course, that's weird on so many levels. It's weird in terms of fundamental thermodynamics, right? How are you actually making more mass from less mass? The one answer I can tell you is that you never, we're not violating thermodynamics here. There's not something weird going on underwater here. But we didn't have a good explanation for how this structure comes about. We tried a couple different hypotheses to explain this. And one of the reasons I was so intrigued by this odd structure is that some folks that first described these inverted trophic pyramids in different sections of our global oceans we're describing them, finding them often in pristine places, less fished places, and we're describing them as potentially a kind of native architecture, a pre-disturbance architecture that was only found in these unusually undisturbed spots. And so I wanted to, sort of get deeper into that, both understand how this may construct itself and then push a little bit harder on whether or not that is truly a native structure we should be looking for in ecosystems. So we tried some complex explanations, some simple explanations. The simple one first, we thought, well, hell, are we counting sharks wrong, right? And that's a real possibility, right? A lot of the tools that we develop for doing marine ecology, the exciting thing about ecology generally, particularly ecology underwater, at least in ocean systems, is it's quite young. You know, we're talking about a science that is arguably half century. Most people who are practitioners before that are dead, so I can say that probably. But it's arguably like really got going about a half century or so ago, right? So these tools are new, but they were also developed and applied in places that may already have been damaged. And so we personalized these tools to the disturbance context where we were actually taking data from. What I mean specifically here is that perhaps some of the survey tools, and there's a very precise methodology, a whole package of methodology you can pick up and potentially use for counting fish on reefs. As it is in science, right, you can have an evening full of beers and conversation about which methods are better and worse, and because there's a very deep literature and a methodology on what is the right method to count fish on a coral reef in a shallow water coastal ecosystem. Our first question was, did we develop these tools in places like Hawaii or Florida that already had lost all these large predators, and so were they not really working well in a place that had lots of predators? One obvious reason that may not work is that in a reef like in Florida where there maybe aren't very many jacks or sharks, that a tool works just fine for surveying less mobile critters, but when you end up with big and very highly mobile critters, maybe that tool doesn't work as well to capture them as they use more space in front of you as a surveyor. So we counted sharks, in this case specifically sharks, underwater using a bunch of different methods, and then we built a simulation where we said, okay, let's create an electronic diver in a simulation and some electronic sharks based on real parameters for ways that sharks use space. Let's give that electronic diver 3 different methods, common methods using what's called belt counts and point counts. I won't elaborate on the methods and video surveys. And let's see if you do in the simulation space exactly what you do underwater and sharks do exactly what they do. Do you overcount them? Are we, in essence, overcounting the number of sharks and so producing this false view of an inverted trophic pyramid? Well, we are overcounting them. So the dotted line here, the nice thing about a simulation that you never will know in nature, in a river and a coral reef, is that I know exactly how many sharks are in my little artificial electronic reef. So I can tell you just how well that those survey methods are operating in that simulation artificial reef. By and large, we're overestimating sharks with all of our techniques. We can figure out actually which of these work better and which of these work worse. That's helpful for counting sharks in the future. But we're not overestimating sharks enough to be able to account for the weird structure. I still have some other explanation that needs to be divined to be able to explain why there are so many large predators or a large biomass of predators in this weirdly structured reef. So the next level up in complexity that we probed was trying to describe or exploring the hypothesis that, as I said, large fish, sharks use more space. They connect more of these sub-communities that may make up a composite of an ecosystem around an atoll or an island. If they use more space, maybe they're connecting more of these different communities. Maybe they're drawing resources from these different communities. Maybe when I go into the reef and I count sharks and I count herbivorous fish and I count prey fish, I'm seeing what's there stacked up in one moment of time in one section of space, but I'm missing some of the connections that a really mobile consumer is making to these other systems. To be able to answer this question as to whether some of these predatory fish were in fact grabbing energy secretly, if you will, cryptically from some of these other systems, we used stable isotopes, natural abundance stable isotopes. I won't go into some of the mechanics. Suffice to say, it's an interesting tracking tool. As I was talking about, you get in the tissue of something like a manta ray or a shark, and the ratios of things like carbon stable isotopes tell you something about where they're eating. In this case, carbon stable isotopes are helpful because carbon that is produced close to coasts is very positive. Carbon that ends up being produced and generated, basal carbon at the bottom of food webs in more offshore systems, more pelagic systems, ends up looking more negative. So you get this gradient of sort of positive to negative basal energy or basal food that is there potentially in the buffet in front of a shark that you're just seeing and you're just counting in this weird inverted pyramid on the reef. And you can look into the shark's tissue and figure out, well, is there anything we can learn about how they may be sampling this isotopic space, this energetic space? So we first isotopically type the different habitats by looking in the isotopic composition of low mobility predators that hang out just in that space. So you go and you grab something that unambiguously belongs to the pelagic system, a tuna. No one debates whether a tuna is hanging out in a lagoon. You grab the similar analog for a reef system or a lagoon system. And I'm kind of modularizing these into three different communities. You have the pelagic system, the reef system, and a lagoon community. So you type what carbon isotopes, what basal energy ought to look like in a lagoon community, a reef community, and a pelagic community with some air around that. Then you go and you grab the predators, all the stuff that makes up that blue block in your inverted trophic pyramid. You sample them, and you run that all through your mass spec, and then you plop them into this same plot. Now, your hypothesis is, okay, so if I'm counting them on the reefs, if I'm assigning them to the inverted pyramid that we're assembling for a portrait of structure for the reefs, if they are feeding exclusively from the reefs, they ought to perfectly overlap with the signals for reef energy, for reef isotopic carbon that I'm sampling only on those reefs. So we can throw them in. I have 2 sharks and 1 very curious, often snapper that you throw in. And we see immediately that on both the x-axis in terms of carbon and slightly less informatively in this context, the y-axis in regards to stable isotopes of nitrogen, that there's not perfect overlap between our sharks and between our reef signal. We can do better in terms of making predictions for where energy may be coming from using some modeling techniques, some sort of simple mixing models. These are results from Bayesian isotope mixing models that take the signature of the sharks and the signature of each of our 3 sources and make you some predictions with some error about where the biomass of a shark is being built. It's different for our different players in that blue block, for our big predators. Here's a story, for example, for the gray reef shark. Numerically, the most abundant shark, numerically the most common shark in that blue block, sitting on top of that inverted trophic pyramid. Quite interestingly, this shark, the model is telling us, is drawing over 80% of its energy from a pelagic system, and only 20% of its energy is actually coming from the reef where we're counting it, where I'm assigning it to structure. So it seems, in the case of the gray shark, that there was indeed this cryptic linkage that was going on, that there was this really important hidden part of the ecology that I wasn't seeing because I was just looking and building a structure portrait, as often we do when we go to a coral reef or when we go to a forest or we go to savanna or you go to a river. You look in one space and you build a portrait of biomass or structure for that space where you're studying. Well, that didn't work so well for doing that on a coral reef because I was missing some really important interactions that helped to explain an important energetic linkage that helped explain an important element of the structure here. The answer is different and variable. So black-tip reef sharks are much better behaved reef sharks. They're hanging out on reefs. And snapper are somewhere in between. This particular species of snapper is somewhere in between. Many people sort of were, including me, surprised by this. Like, this is called a gray reef shark, and it's a bad reef shark. It's called a gray reef shark because our historic understanding of this species was that it belonged to a reef. It fed on a reef. And here we are seeing this chemical signature that it, in fact, belongs in a reef, but it is also pulling energy from a different system. So this helps add a little bit more parsimony to our explanations for thermodynamic structure here. In essence, we're not subverting the laws of how the universe works. We're just having sharks that are going off the radar screen and feeding elsewhere, grabbing energy from these neighboring systems and building their biomass, and then coming back into these systems that we're assaying and counting fish in. So that was comforting to be able to sort of make sense of that. It is always a little, you always have confidence in these results, but when you're using something as indirect as chemistry of tissue to tell you something about what a shark is doing, you wish you had more direct lines of evidence. So we tried to corroborate some of these this chemical insight we were getting from stable isotopes using some direct insight from the shark stories. So we stuck transmitters on the sharks. This is a GPS transponder that has a lot of the same sort of basic technology that lots of you are using when you're tracking terrestrial animals, same kind of technology that is in my phone. When it breaks the surface of the water, it communicates with the satellite and gives me a point of where a shark is. We very happily saw that the tracking data was reinforcing the chemical data, the stable isotope data. Very happily, it seems that this gray reef shark, at least in this place, was a crappy shark, reef shark, that is. It's a good shark. Oh, it's a bad reef shark. Most of the time, it's a good shark. A lot of the transmissions that we were getting were from the project system. Now, you don't get from this tracking data information about precisely what they were doing in that space, but it's highly unlikely that there were a high density observations that were many kilometers from the reef edge that aren't related to foraging. And in some cases, we saw very long forays for particular individuals that were taking them on almost 1,000-kilometer journeys in this totally hidden way outside of this reef. So this is really useful for thinking about structure. Just as an aside, it's also very informative for thinking about management. You know, we're looking to keep these pieces on the chessboard, right? And well, getting some sense of their spatial biology makes us do that better. One of the things that we learned was this gray reef shark, which is a flagship species in this coral reef and this national wildlife refuge, was going well beyond, many of them, going well beyond the boundaries of that wildlife refuge. So what we thought we were protecting by putting a protected area around this ecosystem was insufficient in terms of its spatial extent for protecting the biology and the ecology of this species. As an interesting aside, because some of you we were talking about the sort of modern interplay between our role as biologists and people who are talking about policy. You know, I set out to create this plot to try to answer and feel good about the stable isotope results we got from chemistry. But when we learned that there was a lot more mobility happening from these gray reef sharks and these parks weren't, these specific parks weren't quite large enough, we took this, well, a slightly more elegant version of this plot, and sat down with people from the White House and had a conversation about whether or not this particular marine monument needed to be expanded. It was one of a bunch of different pieces of information that scientists brought to the table that then built a case for the expansion of a neighboring monument. And you know, that data added up to become the world's largest protected area in the oceans, our United States' largest protected area, but genuinely the largest protected area, fully protected area in our global ocean. So it's kind of a cool, you never know exactly what the utility of your science would be. Again, a pure scientific question for me, but we need to track where some of this pure science can play a role in shaping management. Okay, conclusions from story one. Top predators, sharks, others, create cross-system connectivity. They're grabbing energy, grabbing resources from other communities. This connectivity needs to be visualized chemically, sometimes electronically, to be able to interpret the structure of the community that is under study. And then this side, that understanding something about this connectivity it feeds back into how we actually think about safeguarding it and the role that it plays in controlling structure and dynamics in a system like a coral reef. Okay. A new species. We're going to go from a big predator to a big herbivore and talk a little bit about ecological uniqueness. I like big herbivores, land or sea. And one of the things that I think is really interesting to me is whether some of these big chess pieces can be replaced in the jobs they do and the functions they perform in ecosystems by some of the other smaller bodied organisms that may still remain extant after we do our thing to some of these spaces, terrestrial or marine. I do some work in East Africa where we try to experimentally manipulate and create scenarios on landscapes where that mimic the loss of large herbivores by putting up large electric fences that look a lot like Jurassic Park style fences that experimentally remove. They feel a lot like Jurassic Park fences when you wake up too early in the morning and you run into them. but they effectively remove large herbivores like zebra, elephants, others, and then inside that space you have essentially this space that has lost these big actors, and you can look at some of the implications of their loss on all kinds of attributes from plants to reptiles. One of the things that we find quite interestingly in these exposure experiments is that this is an herbivore. One of the things we've kicked out of our experiment, a large an elephant. We also have lots of small herbivores, herbivorous small mammals, many of them rodents, that are eating, interestingly, many of the same kinds of foods that an elephant is eating. You have a mouse, in effect, competing for some resources with an elephant. But there are a set of resources that only elephants are interested in. And interestingly, beyond that, there are only some things that an elephant can do to an ecosystem mechanically that are not replicated. This is an example. It is pushing over a tree. These herbivorous mice I'm telling you about, no matter how much coffee they have in the morning, they're never going to push over a tree. So there are a set of functions, is what I'm saying, that we've learned in this terrestrial work that are truly specific to these large players, either mechanical functions or ecological functions that are not replicated by smaller bodied organisms that remain in these systems. Let's get back underwater. This is a species that the BBC, I thought perhaps appropriately, called an underwater elephant. This is Bulbentopan muricatum, the world's largest parrotfish. I mean, it's the biggest parrotfish by a long shot. This thing is like 1.3 meters, 40 kilograms, or thereabouts, about the size of a middle school kid, or a little middle school kid. I was a little middle school kid. Its large size allows it to do some interesting things. It certainly has a higher rated intake to sustain its own large biomass, but it also does some really interesting things mechanically. So because it's big, it can create a lot of bite force, and it exerts that bite force by literally pulling off, by consuming pieces of this concrete, calcium carbonate, stony coral. Sometimes that's dead coral, sometimes that's live coral, right? So the very big body size of this underwater elephant allows it to do mechanical things to a reef that, at least my starting hypothesis was, were truly unique. So to try to get at how unique this function was and try to quantify and get my head around some of the diversity of functions that perhaps this large species delivered, we did a sort of portfolio of work on the ecology of Bulbentopan, the bumphead parrotfish. The thing I should add is that it's a species of risk. It was considered for listing as a U.S. endangered on the U.S. endangered species and its list, and it's been declining throughout the Pacific. So there is potentially some unique functions, and there's also this important applied value of trying to understand what those functions are, because they are likely going missing along with the fish in a lot of places. We looked at a lot of different kinds of impacts, basically tried to follow the fish and learn everything we possibly could while we're doing that. So we measured rates and targets of consumption, the sort of morphology and mechanics of the feeding, the rates of excretion, and the influence on coral recruitment. We found some really interesting things. As we followed and took this data, we tracked the fish, we tracked ourselves, and hung out with fish. I've never been more fit than trying to swim with parrotfish for several summers. But the tracking of this fish allows us to make spatially explicit renderings of the ecological information that we collect by watching the fish do its thing on a reef. Here are some of those. In aggregate, you put some numbers on what you anticipated was a very powerful source of ecological influence. And here are some of those numbers. One individual, a bumphead parrotfish, in 1 year eats over 2 tons of living coral, which, as you can imagine, is a major source of predation, a major source of change for these foundation ecosystems, for coral reefs themselves, and is genuinely unique. The capacity to bite off these, again, rock-hard corals is in this quantity, at least, in this quantity, removing tons, is truly unique amongst the biodiversity portfolio of Pacific reef fishes. There's 3,000 or so Pacific coral reef fishes. There's about 35 parrotfish. This is the only thing in that entire portfolio that gets anywhere near ton, and nothing else even has a ton of living coral extraction, simply because of its large size and its capacity to actually physically break off and consume these corals. Then we can go to simulation, as we talked about, to maybe extrapolate a little bit of this in situ observation to longer-term patterns about what this predation on corals might mean. The simulation it has a bunch of input parameters, basically where we put coral and algae and parameterize them with some very simple rules. We've taken the feeding preferences that we observed in the field for the parrotfish, put them into the simulation. We've given a size to the coral and the algae, and then we just let them do their thing. We have simulations with no bumphead parrotfish, and then different densities of bumphead parrotfish. Lots of things you can track in your little electronic play reef. One of those is functional diversity. So we first run a simulation without parrotfish. We're just curious, what does the aggregate influence based on what we saw by following all these parrotfish and their feeding preferences and the rates of intake, what does that do to diversity? We see without the parrotfish, we see sort of a moderate decline in diversity. That's because they're not there eating up algae that competes with some of these corals. Then we put parrotfish in medium and high densities, and we saw something kind of interesting. It turns out, maybe not surprisingly, that this at-risk fish is not great, at least in terms of diversity for the reef. Perhaps not surprisingly, a really powerful predator can exert a strong influence. In this case, the actual preferences of coral that they were eating was driving down diversity on this reef. It was kind of a puzzling observation to make sense of, because here it is an endangered species. But we manage for lots of things. Often we manage for diversity, trying to make sense of exactly how to tell that story and exactly what that meant for coral reef health. It's important to always say, okay, look, I just have one of 3,000 coral reef fish in this model, and that just gives me a clear view of what this one fish is doing. We want to know more about that complexity, but it is quite interesting to think about this strong influence on diversity. But as we sort of expanded it, it also influences size of corals. Green guys are algae, the other coral, or the other spheres, the colored spheres are corals. It reduces the size of the coral colonies in our simulation. As we expanded our work, trying to figure out, well, what is the diverse portfolio of influence, we found also that a complex, ecologically influential species like a bumphead parrotfish did lots of things. It ate a lot of coral, but also in the same genre of being able to have this strong mechanical engineering influence that allowed them to eat coral, it also changed reefs in sometimes very positive ways. One of the ways that we discovered as we got deeper into this was that the divot bites that it was making in these reefs kind of look like if you're golfing, at least you're golfing like I golf, you create a lot of divots. It looks like that on a reef, right? When this big parrotfish comes down and takes a bite. It turns out that when that happens on the dead reef, that creates these wonderful little microrefugia, these little nurseries for baby corals, for planktonic corals that are settling on reefs to set up shop. So, as we got more into understanding again, the portfolio of functions, we saw some things that would be, from a manager's point of view, positive, and from a manager's point of view, negative. My collective view is that, wow, first of all, there is something very unique about this herbivore. This is something that's going to be irreplaceable. There's nothing else on the reef that can, if this species goes missing locally or globally, that's going to slot in to do that. But this influence is complex. It's varied. So you see, again, if you're a coral reef manager for this Fish and Wildlife Refuge, you see some good things and some bad things. For this, whatever it is, it's powerful. That seems, and irreplaceable seems to be my bottom line from examining the functional ecology of the species. Okay. I want to talk about ecosystem ecology in the last quick story here. We've talked a lot about mouths. I want to talk about anuses for a little bit. And this is still a story of connectivity. So you have things like sharks that are predators and consumers that are interactively linking. Again, think of this as maybe our reef and our pelagic system. You have things like sharks that are building these bridges between these high-density nodes of ecological interaction, which shape these dynamics in important ways by feeding back and forth between these systems. But they're also pooping back and forth between these systems, right? And those transfers of energy, those transfers of nutrients, could be quite consequential ecologically. So the hippopotamus, naturally. We started working in a hippopotamus because they seem to be a very good candidate study species for examining these interactions because, again, they fit this profile of a large-bodied species that's highly at risk. Hippopotamus somehow don't get the kind of airspace that many megafauna in Africa get in terms of their declines. They are declining. They're listed as vulnerable species all across sub-Saharan Africa. Major range contractions in near and far ecological time for hippopotamus, and they fit the bill certainly for this potential connector of transfers for energy. They eat, so they're amphibious. Everyone probably knows this, but hippopotamus is an obligate amphibious mammal. It has to be in water, but it hangs out in refuges in water during the day, and it goes out at land at night and feeds and goes back and forth, right? It eats tons, literally tons of organic matter on land, and then comes in and poops tons, literally, in aquatic systems. And you know, sometimes your dissertation is just the recapitulation of a nature documentary, and that's what this is sort of about, right? It's about trying to demonstrate what is obvious in this film, is that those transfers are quite consequential for aquatic life in streams and lakes that these hippopotamus interact with. Correct me if I'm wrong, but I'm going to make a wager that that's the first time that in your class, in the lecture, you have seen a fish anus hippopotamus interaction. Am I right? Okay, perhaps not the last, but so there are lots of ways to get at various different attributes of the importance and the consequences of the loss of this large consumer. We can use isotopes again. I'll show you some of that. You can visualize where they're moving, sort of like the analog of the shark, but with the hippo. We look at and are quite interested in how these transfers shape water chemistry. We're quite interested in how those changes in water chemistry change ecology. You know, we saw fish. It certainly is a strong influence in all attributes from producers, macroinvertebrates to fish and rivers. We also do some manipulations where we kick hippos out of riparian systems. I'm just going to, in the interest of time, showing you a couple of the top things. The first is like the hippopotamus version of the shark story. I was curious, and you know, you're sort of aware that there's a lot of this eating and pooping going on, but does that matter? Do these transfers of sometimes limiting nutrients that are going from terrestrial systems to aquatic systems actually matter? Are they being utilized by aquatic consumers? So you here isotopically type some of your energy sources. I'm using carbon again as being very influential and informative. You have C3 trees that have a unique signature, and then C4 grasses and hippopotamus dung that have their own kind of signature in carbon isotopic space. You grab a handful of aquatic consumers and you look to see if you see evidence for dependency, evidence for sort of a strong imprint of the isotopic signature of hippo dung in the tissues of some of these potential consumers. Unlike a coral reef, this is a very physically dynamic place. Naturally so. We have a pretty dynamic dry season and wet season in this river, and then sometimes it dries entirely, or it dries except for a few refuge pools. This is actually what it's looking like right now. This river is dried down in central Kenya to a state like that presently. Surely that hydrological forcing, surely that physical context changes the impact of these subsidies and how animals in this space use those subsidies. We're trying to keep that in mind, and I'll show you results with that in mind. I'm going to plot dependency for just one particular kind of river fish during the wet season and during the dry season. We're going to look at some comparative results from pools for fish that were sampled in pools with hippos and pools without hippos. Hippos are pretty compliant methodologically because they're gregarious. They end up like to hang out in one spot. So you have hippos year after year, after decade after decade, going back to the same pools in a river. Anyway, I'll show you what they look like in carbon isotope space. We took a bunch of fish out of the river, put them into captivity, fed them just dung. Sounds like a dream. Wouldn't you like to be that experimental unit? Fed them just dung, and then looked at the isotopic signatures of their tissues to see, okay, what does it look like if you just eat hippo dung? They turned out they loved it, these fish. That gives us a sense of like what's the isotopic directionality that you should see change happen in pools with and without hippos. So during the dry season, we look at the signatures of these fish. We see some tendency for increased isotopic sort of a presence of hippo-derived material in our fish with hippos, but during the wet season, that's not a significant difference. When we go back to the same space, same pools, same species of fish during the dry season, when these nutrients are concentrated, when they're less diluted, we actually see the same pattern, except it's now more significant, it's now a significant period, where we see fish in hippos, in pools with hippos, look like they're doing what the video said. They look like they are definitely deriving energy, they're definitely building biomass based on utilization of these subsidies, the dung that hippos are bringing into these rivers. So there is that dependency you're able to visualize chemically, and then this important sub-story about how physical changes in this hydrologically dynamic river shape the implications of how those subsidies are utilized. Drying and the wet-dry cycle of this particular river happens over the course of the year. There's also some, of course, as we know, very profound implications for long-term hydrologic dynamics in East Africa, in Utah, everywhere else, as a result of climate change. This is sort of preliminary patterns already manifesting themselves in East Africa, where we see in long-term hydrological records starter evidence of patterns suggesting of longer and deeper drought, more intense storms, and these reduced water flow events over time in the river. So in the here and now across seasons, we definitely see an influence for how ambient physics shape the implications of the impacts that a big animal has in terms of a nutrient vector on the recipient system. Very likely that we also need to be thinking ambitiously about how the role of these hippos is going to be sort of re-engineered in a new hydrological future for these same systems. Okay, so a very quick bit of just a sneak preview, I suppose, of what we learned from tracking hippos. One of the things I learned first, nobody's ever tracked hippopotamus. I learned exactly why nobody's ever tracked hippopotamus, because it's really, really hard. But we figured out, in fact, a grad student named Tristan from UC Berkeley and another postdoc in my lab, both of them, we've been working together for a long time, have figured out how to track hippopotamus. This is some of Tristan's work, where he's using a crossbow, placed a sensor on the back of a hippopotamus, and we're getting short-term tracks of where the hippopotamus are going, the first in the world, that you're getting the first view of where a hippopotamus moves across the landscape. This is, of course, useful for thinking about the pure spatial biology of the animal. It's very useful for thinking about these transfer dynamics. I can, and Tristan does, go through and create these nutrient shed maps using information about both what space and what information we're getting from other sensors on these tags that tell us how they're using that space, to be able to actually plot where energy is being grabbed and where it's being deposited, as well as building out profiles for how it's using space. So this spatial information is good for trying to link space to chemistry to understand the important role that these large consumers play in shaping community or shaping ecosystem ecology. And they're also, of course, useful for thinking about raw management of a at-risk species, right? How are you going to manage a declining species like a hippopotamus if you want to keep that particular player on the chessboard if you don't know how much space it needs? So they're good for that. Takeaways from last section. They're, in this case, a very iconic example, a powerful example of how large animals eat a lot, move a lot, poop a lot, and shape nutrient dynamics across ecosystems. Some first evidence of how those transfers can be ecologically consequential, and then this sort of powerful interplay between spatial ecology and ecosystem ecology that helps us actually visualize where all of that is happening in a landscape. So returning, kind of to where we started, there I think is a lot that we've begun to learn, a lot more that we can learn about how body size shifts, particularly losses of larger constituents from ecosystems, change the structure of systems, and how those changes to structure influence the dynamics of systems. It seems that this change is not endemic to predators, large-bodied predators, but is also something that's important to large-bodied herbivores. This insight that we collectively harvest from the important role of these large consumers helps us, I think, to get a better handle on how the modern systems that we're working with, surveying, trying to interpret, actually operate. In systems like Utah, in systems like Africa, where we're losing a lot of hippopotamus, it gives us a good sense and intuition, sometimes quantitative, sometimes qualitative, about legacies of past change as a result of loss of these megafauna might be. And importantly, it gives us some important predictions for future change. Again, a last visitation to this metaphor of the chessboard. If we're removing all these large pieces, it seems that global change, climate change, is actually also changing the placement of all the squares on the chessboard. So just as we begin to get a handle on what it means to remove the large-bodied species from our systems, we're actually changing the very game, the very structure, the way the game is played. We're changing the underlying physics of many of these systems. That makes interpreting what the loss of big animals, large-bodied consumers, means from systems more complex, more difficult, but it also makes it more important endeavor than ever. So I will leave you with that thought. And if you have any questions, I'd love to answer those.
A couple of minutes for questions.
Yeah.
So for the sharks that are, traveling, what's the benefit for them to travel? Like in terms of optimal foraging theory, why would you go off the reef to eat?
Yeah. So the resources are quite good when you can find them. In a pelagic system, you know all this, but in a pelagic system, I often think of it as kind of like a desert with a bunch of oases. Oases being like big schools of bait fish that if you can find them, you can cash in big time. The reef, from a shark's eye point of view, is a really important source of food, but it's always there, but it's really hard to catch. We've spent hundreds of hours underwater on reefs. You've probably spent thousands of hours underwater on reefs, and the number of times that you see a shark actually catching and eating a fish are incredibly rare. Like I've seen that happen once, right? So it's really hard because it's really an architecturally diverse system where it's a wonderful place with micro and macro averages that a prey fish can get away from a shark. The advantage for this consumer, that it has the morphology and the physiology to allow it to go off a reef, is that if it can track and find those oases, it catch them big when it can find them. And then it comes back to potentially the stable system where it can otherwise refuge, aggregate, and feed. It definitely feeds on the reef, but you go back and forth between these different kinds of energy pools have their own sort of, from optimal foraging theory, have their own pluses and minuses. Yes?
A related question. So you get the same signature if the pelagic fishes were coming inshore to refuge and then get whacked.
Precisely. Right.
Any indication that's happening in the system? Exactly.
So if you had, I didn't put a picture in, they do feed on these small clupid-like sardine-like fish. So there's two answers, potentially. Just looking at the isotopic plight, I can't tell you whether the pelagic fish are coming onto the reef and the sharks are going up and grabbing them, whether the sharks are going out in the pelagic system and grabbing them. The tracking data helps, so that when I see them spending, some sharks, 90% of their time off the reef, that builds my confidence that it's more the sharks going to the food rather than the food coming to the sharks. But both can happen, and probably both can shape the potential for this inverted trophic pyramid structures. It doesn't really matter. This cross-boundary flow of energy can happen by virtue of the spatial volition of the shark or by the prey coming in. That's a good point.